Hello everyone, and welcome to the second lecture in our Introduction to Forensic Science unit. Congratulations, you have made it past your first one. I am very proud of you. This one's going to be on the scientific method. I know you have probably learned this in every single science class you've ever been in, but we want to keep it fresh in your mind, so we're going to review it again. For me, I have the scientific method broken up into 5 steps. Sometimes you'll see 6 or 7. It doesn't really matter the number of steps that you have. It's really the process that's important. And for the most part, it's the same process, no matter which version you use. For me, step number 1 is I'm going to observe the world and I'm going to ask a question about it. So fall is going to be coming pretty soon, so I might notice that the trees are changing colors, and I will say to myself, hey, I wonder why the trees are changing colors. Okay, so I have observed something, and then I have asked a question about it. And then after that, you might go, well, what are some possible answers to that? Right, and that's when you're going to do step number 2. You're going to make a hypothesis. Right, and your hypothesis is basically what you think the answer to your question is. It may be right, it may be wrong. You're going to do an experiment to find out what that is. So that's step number 3. You're going to conduct an experiment. So you do your experiment, and then you get some data based on your experiment. So step number 4 is going to analyze that data. And then once you've taken a look at the data, you're going to try to figure out what it all means. So was your hypothesis correct or not? So step 5 is you're going to make a conclusion. Now, when you're setting up an experiment, you need to keep a couple of things in mind. The first are variables, and all a variable is just something that can change in the experiment. And when we do the experiment, we want to look at one variable at a time. Right? We do this because we want to make sure it's only that one thing we're looking at that's causing something to happen. If you use 2, you may not, be sure which one, right, caused the reaction you were looking for. Now, in an experiment, there's going to be 2 different variables that you're going to look at. Okay, one is the independent variable. Okay, and this is the variable that you are testing. Okay, that one thing that you're focusing on to see if it's, causing your observation or not. All right, and this is the one that you manipulate. All right, you actually change this one. Okay, this is what you're going to set at different levels and things like that. All right, now the other one is the dependent variable. And it's called a dependent variable because this variable depends on the independent variable. Right? This is the thing that or the thing that changes because of the independent variable, because of what you're testing. And this is what you're going to observe, okay, or to measure. So once you've figured out what your variables are for the experiment, then you're going to go ahead and you're going to set up your groups. And there's 2 different types of groups you can have for an experiment. The first is the experimental group, and the second is a control group. Right, now the experimental group is going to get whatever you are testing. Flip side, the control group will not get what you're testing. And the control group is going to act as our comparison. So for the experimental and the control group, we want to make sure that we try to keep the conditions the same for both groups except for what we're testing, or as close as you can possibly get them to being the same. And that's to make sure that it's only what you're testing that's causing the change. Okay, or giving us the results that we have. So you're going to watch a very short demo with soda cans. So let's play that. So we made some observations about these soda cans, and now we're going to go through how you would use this for this scientific method procedure. So I made the observations about the cans. The diet can was floating, and the regular can of soda sank to the bottom. Okay, so immediately, the first question that comes to my mind is, why did the diet soda float and regular soda sink? And then I'm going to start to think about answers to that question, which would be my hypothesis. So if you take a look at the cans of soda, diet soda lists no sugar, and regular soda lists like 24 grams, which is quite a lot. So I think it's the sugar that is causing this change. So for my hypothesis, I'm going to put that cans with more sugar will sink. Down more in the bin. Okay, so I mean that's pretty specific. I can test this. You want to make sure that your hypothesis is testable. And I'm pretty sure I can test this. So if I have cans and I increase the amount of sugar in it, I should see that the can should move down in the water. So now I'm going to focus on the groups. I have got my hypothesis. Now I'm ready to do the actual experiment. Right, so I'm going to set up 2 experimental groups for this. You could set up more. And I'm going to set up my control group. For my experimental group, these are the ones that get what I'm testing, which is the sugar. That's what I want to see if it caused the change or not. Right, so for my first experimental group, I might have my can, let's say filled with a certain amount of water, and it has a higher level of sugar. Now, I would want to be specific about what these numbers are, but for right now, we're just going to use it as an example. Okay, for the second experimental group, I might have the can with the same amount of water plus a medium sugar amount. And then for the control group, that does not get what I'm testing, so this would be a can that does not have any sugar in it at all. Okay, but the cans and the water, we're going to keep the same. Because we want to make sure that it's just the sugar amount that's causing this change, and not, any of that other stuff, any of those other variables. So we're focusing on one at a time. So we did our experiment, and let's say that we got these results. So I had my first experimental group. Let's say I had 20 milliliters of water that I had in all of these cans. So, the water is kept consistent all the way through. All right, but for my first experimental group, I wanted a higher amount of sugar, so I chose 10 grams. Okay, and I tried this experiment out 3 times. All right, you want to do an experiment more than once to make sure that it wasn't like some sort of random fluke. Okay, so when I did that, I found that the distance of the can from the bottom of the tank, so if this is your tank, this is the bottom. right, we're measuring how far the bottom of the can is to the bottom of the tank. Right, so when I had the high amount of sugar, the bottom of the can was right at the bottom of the tank, so it was 0. Right, for my second experimental group, we said we wanted a medium amount of sugar. All right, so I chose 5. And if we took a look at this group, all right, it was, about like halfway up the tank. All right, and then for the control group, when we had no sugar at all, these cans stayed towards the top of the tank. Right, so that's the data that we got, and now we're going to make a conclusion based off of it. Right, so you can see the ones that had more sugar, okay, stayed towards the bottom of the tank. They sunk more, and the ones with less sugar stayed towards the top. Right, so I can say that my hypothesis had support. Yes, if I have more sugar in the can, it is going to sink farther down in the tank. Okay, so my hypothesis has support. Yay! Alright, and sometimes your hypotheses won't have support. Sometimes you'll find out you were completely off the mark. And that's fine, because that told you that it wasn't that thing. Now you should go test a new thing. So it rules out possibilities for you. So negative hypotheses for scientists are just as important as positive ones.